Hello everyone, welcome to the Bodhi Academy. In today's video, we are going to continue with Standard 12 Chapter 11, that is Biotechnology Principles and Processes. So, in the last video, we have learnt about how the restriction endonucleases or the restriction enzymes while cutting the DNA fragment forms the blunt ends and the sticky ends. Then we have also learned about how the nomenclature of this restriction endonucleases is done. So let us begin with a new video. Biotechnology Principles and Processes. The steps in formation of recombinant DNA by the action of restriction endonuclease enzyme, that is E. coli. So let us see how the DNA fragments are cut by the restriction enzyme. Now this is a vector DNA, the vector plasmid, and this is the foreign DNA which we want to introduce in this vector DNA. So let us see, the enzyme cuts both the DNA strands at the same site. Then E. coli cuts the DNA between bases G and A only when the sequence GAATTC is present in the DNA. So first of all, the enzyme will cut both the ends of DNA at the same side. So here we can see it is cutting at cutting between G and A. Here also same. Here also between G and A. Here also it is cutting at the same point. Then now the E. coli cuts DNA between G and A only when the sequence is this GAATTC, and we can see here the sequence is GAATTC, as well as over here it is the same sequence. Then after cutting, now this will form a sticky end, that is one strand of DNA will be a little longer. Okay, so this slightly longer strand of DNA will form the sticky end. Same for this DNA, that is foreign DNA. Now what will happen? Now we have to join these two DNA fragments. So the DNA fragments join at the sticky end. So this part will go and fix over here. Okay, so these sticky ends will join together to form the recombinant DNA. Recombinant DNA means this modified DNA in which we have added the gene of interest or the DNA of our choice. So this joining of the DNA fragments is done by the enzyme ligase. Then after this, okay, so here in this diagram we can see that here we have a foreign DNA, the one that we want to introduce in the organism, and this is the vector DNA plasmid. We will cut a portion of this DNA, foreign DNA, and introduce it in this vector DNA. So first of all, we need to use the same restriction enzyme to cut both the fragments. Okay, so same restriction enzyme cutting both foreign DNA and vector DNA at the specific point. So we have to use the same restriction enzyme to use both the DNA fragments. Then see how the DNA has cut this fragment. So here we have a small part of DNA which we want to put in this vector DNA, and here also the cut has been made by the enzyme. Now what we have to do, we have to transfer this fragment to this vector DNA. Then after that the ligases join the foreign DNA to plasmid. So the foreign DNA will be joined to the plasmid DNA by the ligases. Ligases means those enzymes which join or link the DNA fragments. So as we can see, the blue color DNA has been added or joined to this vector DNA. Then after this we have to introduce this vector DNA to the bacteria or the cell. So here we have taken the E. coli cells, and then this process is called as the transformation. Then after the cell divides, this DNA which we have, the recombinant DNA, will be found in both these cells. So this is the diagrammatic representation of recombinant DNA technology. Then recombinant DNA, that is called as the rDNA. Now restriction enzymes are used in genetic engineering to form the recombinant molecules of DNA, which are composed of DNA from different sources of genome. So as we can see, this DNA, the recombinant DNA, this recombinant or rDNA, this rDNA is formed of fragments from different sources. So this is one source and this is the second source. So it is formed from sources from different sources. Then when cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends. Okay, so since the restriction enzyme used is same, it will form same type of sticky ends, and these can be joined together, that is end to end, by using DNA ligase enzyme. Now unless one cuts the vector and the source DNA with the same restriction enzyme, the recombinant DNA or vector molecule cannot be created. So we have to use the same restriction enzyme for cutting both the foreign DNA as well as the vector DNA. So this is all in this video. Hope you have understood this. In the next class we are going to learn about the gel electrophoresis. So thank you for watching my video. Please like, share and subscribe to the Buddhi Academy.